Keep it going.
Okay. And pyrimidines, which is cytosine and thymine. Adenine and thymine are paired. And cytosine and guanine are paired together. This is called base pairing. Each of these pairs are bonded together by a phosphate bond. Complementary base pairing is when you know one strand of the DNA sequence, then you'll know the other strand. Like here, T, G, A equals A, C, T. The template strand, which runs 5' to 3', is anti-parallel to the growing strand, which runs 3' to 5'. So it's like a mirror. The DNA strands are held together by hydrogen bonds. Adenine and thymine are held by 2 hydrogen bonds, and cytosine and guanine are held by 3 hydrogen bonds. DNA is in the shape of a double helix.
Good stuff.
In DNA replication, the helicase unzips the DNA by breaking the hydrogen bonds. This begins at origin replications. DNA strands now form a Y-shaped replication fork. DNA polymerase jumps in at the origin of replication and adds the new nucleotides. Leading strands build quickly and continuously and moving away from the origin of replication. Lagging strands build slowly and discontinuously and moves toward the origin of replication. It is made in pieces of nucleotides known as Okazaki fragments. Ligase joins the Okazaki fragments to make one continuous lagging strand. DNA replication is known as semi-conservative replication. Semi means half or part. And conserve means save.
Edit.
DNA passes its information to RNA. Proteins called transcription factors control the genetic information from DNA to RNA by building to specific DNA sequences. The sugar in RNA is called ribose. Instead of thymine, RNA has uracil. There are 3 types of RNA. First is messenger RNA, which copies the information stored in the strand of DNA. Ribosomal RNA makes up part of the ribosomes, which is produced in the nucleus. Transfer RNA shuttles amino acids to the ribosomes. Now we'll see how RNA directs the synthesis of proteins.
Yeah.
First step is transcription, which involves copying the genetic code directly from DNA. Transcription begins at special sequences of the DNA strand called promoters. Another enzyme, RNA polymerase, brings free-floating RNA nucleotides to the DNA strand. The mRNA strand is now ready to move out of the nucleus, but before it can leave, it must be processed.
1 second, please.
The regions that express the code for the polypeptide are exons. The non-coding regions in the mRNA are introns. Introns must be removed before the mRNA leaves the nucleus. Now our mRNA leaves the nucleus and searches for a ribosome. This process is called translation. The mRNA molecule carries the message from DNA in the form of codons, which is a group of 3 bases that correspond to one of 20 amino acids. Woo! The other end, anticodon, has 3 nitrogenous bases that can base pair with a codon in the mRNA. A peptide bond is a bond between 2 amino acid units. The synthesis of a polypeptide is ended by a stop codon. The RNA molecule carries out the instructions in DNA, producing the proteins that determine the course of life. It goes from DNA to RNA to proteins to July. One type of mutation, which is a change in amino acid sequence, is a base pair substitution. There are 3 types of base pair substitutions: silent, missense, and nonsense. Silent shows no detachable change in the amino acid chain. Missense is a codon is altered and changed the amino acid chain. And nonsense is when the stop codon ends the chain early or no chain at all. You guys are now experts on DNA replication.
You are welcome.
Yay, yay! Bloopers! DNA replication is known as semi-conservative replication. Semi means half or part. The RNA molecules carry out the instructions in DNA, producing the proteins that determine the course of life. I don't know. One type of mutation, which is a change in amino acid sequence. I can't do it. Now you guys know everything you need to know about molecular genetics. You guys are now experts on DNA replication. Oh,